The stroma, or colorless liquid surrounding the grana in the chloroplast, is the site of the Calvin cycle. The Calvin cycle is the light-independent reaction of photosynthesis, which has three reactants: carbon dioxide, ATP, and NADPH, as well as three products: ADP, H2O, and PGAL, which will eventually become glucose. It also has three stages: carbon fixation, reduction, and regeneration. At the beginning of carbon fixation, three CO2 molecules enter the plant through the stomata and are broken apart by the enzyme Rubisco and formed into 6 phosphoglycerate molecules. The 6 PGA molecules then enter the reduction phase of the Calvin cycle. 6 ATP molecules interact with the PGA molecules, contributing energy to them by having one of their phosphate bonds broken. 6 NADPH molecules are also involved in the reduction process and lose one hydrogen to be broken down into 6 NADP+ molecules, as well as 6 water molecules and 6 phosphates. The resulting 6 molecules are phosphoglyceraldehyde molecules, also known as PGAL or G3P molecules. One of the 6 PGAL molecules is released from the cycle to eventually be used in the production of glucose for the cell. 2 total PGAL molecules are needed to make 1 molecule of glucose, which amounts to 2 turns of the Calvin cycle. In this regeneration phase of the Calvin cycle, 3 more ATP are utilized for their energy, and 3 more ADP and phosphates are produced. This results in the molecule ribulose biphosphate, or RUPB. Just 3 RUPB molecules are produced, and they go on to combine with carbon dioxide entering the plant, as well as the Rubisco enzyme, and the cycle begins again. The light-dependent reaction of photosynthesis occurs in the thylakoid within the chloroplast. The reactants for this process are sunlight, water, ADP, and NADP+. The products are ATP, hydrogen, oxygen, and NADPH. H2O molecules, entering from the stomata, release a high potential energy electron when converted into oxygen. This electron travels first through photosystem 2. Sunlight energy is made of photons, or bundles of electromagnetic energy, which is absorbed by chlorophyll pigments. The energy from the light within the chlorophyll molecule excites an electron from its ground energy level to an elevated energy state. This excited electron's energy is then used to propel a hydrogen across the thylakoid membrane against the concentration gradient into the inner thylakoid. The electron then continues on to enter photosystem 1 and repeats the process of getting excited to a higher energy state, but this time donates its energy to the reduction of NADP+ molecules and H+ molecules to NADPH molecules. These will go on to supply energy to the Calvin cycle. A high concentration of H+ molecules now exists in the inner thylakoid due to the previous active transport that brought them across the membrane. Only passive transport is needed to allow the H+ molecules to leave the inner thylakoid through ATP synthase and enter the stroma, which has a lower concentration of H+ molecules. This action generates the energy to bind to ADP with phosphate, which will create ATP, also used in the Calvin cycle.
In extremely hot and sunny environments, plants can often revert to a different process of photosynthesis to reduce photorespiration. One alternative to the C3 pathway is the C4 pathway, which is used by plants such as sugarcane, corn, and crabgrass. These plants live in hotter climates and only make up 3% of vascular plants. In this pathway, the light-dependent and independent reactions are separated, with the light-dependent reactions occurring in the mesophyll cells and the Calvin cycle occurring in the bundle sheath cells. In this process, CO2 first enters the cells through the stomata and is fixed to form a 4-carbon acid named oxaloacetate using a PEP carboxylase. The 4-carbon acid is then transferred to the bundle sheath cells where it undergoes the Calvin cycle using fixation by Rubisco. In the alternative pathway, called the CAM pathway, plants in extremely hot and arid environments such as cactus and pineapples utilize the CAM pathway to reduce water loss from leaves. Unlike the C4 pathway, the light-independent and dependent pathways in CAM pathway are not separated through area, but rather through time. During the night, stomata are left open to allow CO2 to enter. The CO2 is fixed with the PEP carboxylase to be converted into an organic acid. Then during the day, CAM plants keep their stomata closed, but still transfer the organic acids to be broken down into CO2, which is then inserted into the Calvin cycle, as it is done in C3 and C4 pathways.